Welcome to Biochemistry DSP channel. Today I'll be discussing about electron transport chain. It's also called respiratory chain. Before starting with this electron transport chain, you have to know what is oxidation and reduction reactions. Oxidation is defined as addition of oxygen, removal of electron or removal of hydrogen. So oxidation means loss of electron. Reduction means gain of electrons. Or in other words, we can say oxidation by removal of hydrogen. So, reduction means gain of electron or hydrogen. In oxidation reduction reaction, that is called redox reaction, one substrate undergoes oxidation and the other substrate undergoes reduction simultaneously. So, that type of reactions are said to be redox reactions. In biological oxidation, transfer of electrons from reduced coenzymes through electron transport chain to molecular oxygen takes place. So, this biological oxidation refers to cellular oxidation of various metabolic fuels or metabolites by the removal of electrons and hydrogen and addition of oxygen. So, in biological oxidation, ultimately uh, the metabolites will be oxidized to CO2 and water through electron transport chain, and the free energy released during the oxidation will be captured in the form of high energy phosphate, that is ATP. So, this process uh, takes place in mitochondria. Okay, this coupling of oxidation with phosphorylation will be called this oxidative phosphorylation. So, in the biological oxidation, when any substrate such as glucose, citrate, pyruvate, ketone bodies, any metabolites, what actually it will happen means it will undergo oxidation. So thereby it will loses the reducing equivalents, that is electrons and H plus ions, to the cofactors NAD plus and FAD to generate NADH plus H plus and FADH2. So in mitochondria, the reduced cofactors are then oxidized through electron transport chain in the presence of oxygen, so that this reducing equivalents will be converted into NAD plus and FAD. Finally, oxygen will be converted into water. This during this process of electron transport chain, a free energy will be trapped. Okay, the free energy released will be trapped in the form of ATP. That will be utilized for the generation of ATP from ADP and PI. That process is called as oxidative phosphorylation. So this electron transport chain is nothing but the sequence of reactions in which the hydrogen atoms and electrons are removed from the metabolite through a specialized set of electron carriers, and they will be finally help to convert a molecular oxygen into water. This electron transport chain is located in the selective permeable inner mitochondrial membrane. So the enzymes and the components of electron transport chain are located in the inner mitochondrial membrane, which are concerned with transport of reducing equivalents with their final oxidation into water. So this is a structure of mitochondria. So this mitochondria consists of an outer membrane, inner membrane, there is an intermembrane space, and as well as matrix. Okay, so in the inner mitochondrial membrane only, the electron transport chain will complexes, the complexes which are involved in electron transport chain are embedded. So this electron transport chain consists of totally it will have five distinct electron carriers that acts in sequence to transfer electron from the substrate to oxygen. So out of this five complexes, four complexes are involved for electron transport chain, and the fifth complex is called ATP synthase complex, okay, which is concerned with oxidative phosphorylation. Okay, these are the okay, the in mitochondria, the complexes, the four complexes of electron transport chain are arranged in a specific order. They help to transfer the electrons in a specific order. So ultimately, the electrons will reduce oxygen into water. That is the final reaction of electron transport chain. So in during this electron transport chain, what is happening means the electron carriers are sequentially arranged in the order of their increasing redox potential, okay, which are responsible for collection and transfer of electrons and hydrogen from one substrate, and finally they will be helping to convert oxygen into water. During this flow of electron, free energy also will be released, okay. So this energy is conserved in the form of ATP. So electron carriers or the components of mitochondrial electron transport chain, okay. So this electron transport chain has got carriers, electron carriers. These are the carrier proteins, that is nicotinamide nucleotides like NAD plus, and flavoproteins FMN, FAD, iron sulfur proteins FES centers, ubiquinone, cytochromes. So all these act as a carrier of electrons. So this NAD plus and NADP plus are derived from the vitamin niacin, which will be the first component of electron transport chain. So they serve as the coenzyme in the oxidation reduction reactions catalyzed by NAD plus or NADP plus dependent dehydrogenases. Okay. So during this uh, NAD plus will be reduced into NADH by transferring a hydride ion from the substrate like a pyruvate, isocitrate, malate, etc. Okay, whereas this the second hydrogen will be removed from the substrate in the form of free H plus ion. So the NADH will be serving as the substrate for electron transport chain, and loses is reducing equivalent to FMN linked NADH dehydrogenase flavoprotein, which is the next component of the electron transport chain. So like this, all the carrier proteins will be embedded in the inner mitochondrial membrane. So the flow of electron takes place from one carrier protein to the next carrier protein. This flavor proteins, FMN dependent NADH dehydrogenases. So they help to transfer reducing equivalent of NADH plus H plus to FMN, yielding FMNH2. So succinate dehydrogenase is a flavor protein containing enzyme. Then iron sulfur proteins, that is the FES proteins, will be alternatively oxidized Fe3 plus into Fe2 plus during the electron transport chain. Then ubiquinone or coenzyme Q is neither a protein or a nucleotide but a lipophilic electron carrier. It is a lipid soluble protein with which will have number of isoprenoid units. So ubiquinone means everywhere. The term ubiq means everywhere. So ubiquinone occurs in all the cells. It's a mobile diffusible electron carrier. It helps to carry the electrons from flavoprotein and transfer them to cytochrome B. This coenzyme Q transfers electrons from FMNH to and from FADH to the quinone portion of COQ molecule is alternatively oxidized and reduced during the transport of electrons in electron transport chain. Then cytochromes or iron containing heme proteins in which the iron is present in the center. They help to uh, carry the electrons in the mitochondrial electron transport chain. The cytochromes or cytochrome B, cytochrome C1, cytochrome C, cytochrome A and cytochrome A3. These are various cytochromes of mitochondrial electron transport chain. The flow of electrons in the mitochondrial electron transport chain. So during the transfer of electron, the reaction which occurs is one of the substrate will be oxidized and the other molecule will be in the reduced form. So it will have both oxidant and reductant. One will be in the oxidized state, the other will be in the reductant state. So during the transfer of electron, one might be converted from the oxidized into reduced state and the other may change from the reduced state to oxidized state. So the flow of electrons will be through this different carrier proteins like FMN, FAD and coenzyme Q, FES center and cytochromes. So the components carry the electrons from the reduced coenzymes like NADH, FADH to final electron acceptor will be oxygen. The components are sequenced in the order of increasing redox potentials. That is, they are arranged. How the flow of electron takes place means the flow of electron occurs from the increasing redox potential. So in the electron transport chain, these components or the electron carriers will be arranged in the order of redox potential. So they will be arranged in the order of the increasing redox potential and the passage of electrons will be associated with loss of free energy. So during oxidative phosphorylation, this free energy will be utilized to phosphorylate ADP to become ATP. So most energy from the redox. So the how the uh, electrons are sent to NAD plus and FAD means the initial step in the mitochondrial electron transport chain is the generation of reducing equivalents through dehydrogenase enzymes. So these dehydrogenase enzymes occurs in the TCA cycle or beta oxidation of fatty acids or in the glycolytic pathway. So they produce the reducing equivalents. So here NAD plus is reduced to NADH by accepting a hydride ion and the reduced NADH is oxidized by FMN dependent NADH dehydrogenase. So these are the various initial reactions which helps to generate the reducing equivalent. So through glycolysis, NADH will be generated. Through pyruvate dehydrogenase reaction of TCA cycle, we get NADH. and through Krebs cycle or TCA cycle 6 NADH and 2 FADH2 will be produced. So this is how the reducing equivalents will be produced in the mitochondrial electron transport chain. Then electron transport complexes. So four different electron transport complexes are isolated in the inner mitochondrial membrane. So the complexes are arranged in the order, sequential order, so that they help to carry the electrons from one transporter protein to another transporter protein. The transporter proteins are arranged in the order of increasing redox potentials. So the first complex, the name of the first complex is called as NADH coenzyme Q oxido reductase complex. Then the second complex is called as succinate coenzyme Q oxido reductase complex. It's also called as succinate dehydrogenase complex. Third complex is coenzyme Q cytochrome C oxidoreductase complex, and the fourth complex is called as cytochrome C oxidase complex. So these four complexes are involved in electron transport chain, whereas the fifth complex is called as ATP synthase complex, which is responsible for oxidative phosphorylation, that is production of ATP. So during electron transport chain, only these four complexes will be involved in the transfer of electron. So the name of the first complex is NADH coenzyme Q oxidoreductase complex. So here in the first complex, how the flow of electron occurs means from NADH dehydrogenase. It has got NADH dehydrogenase, FES center. So the electrons from NADH to coenzyme Q takes place through this first complex. Okay, so the flow of electron will be from NADH to coenzyme Q. So through FMN and FES center. So FMN and FES center will act as mediator to transport the electron from NADH to coenzyme Q. So the coenzyme Q will be the acceptor for first complex. The second complex is called a succinate dehydrogenase complex, where the flow of electron will be carried from FADH2. So here also the acceptor will be coenzyme Q. Third complex is coenzyme cytochrome C oxidoreductase complex. So this consists of cytochrome B, cytochrome C1 and FES center, helps in the catalyzation of oxidation of coenzyme Q. Then fourth complex is called as cytochrome C oxygen reductase complex or cytochrome C oxidase complex. So these are the four complexes involved in the electron transport chain. So in the first complex, the name of the first complex is NADH oxidoreductase complex. So why it is called NADH oxidoreductase complex means NADH will be acting as the source of electrons. So how the electrons are generated means from NADH. So this will be acting as the source of electron. So from this source NADH, the electrons will be taken up by the mediator proteins or the carrier proteins like FMN. So source of electron is NADH. From NADH, electrons will be transported to FMN. Then from FMN, it will be donated to FES center. Then from FES center, it will be finally given to the acceptor protein UBQ0. So this is the how the flow of electron occurs in the complex 1. So for complex 1, the electron flow occurs from NADH to FMN, from FMN to FES, and finally it will be given to UBQ0. So UBQ0 will be reduced into UBQ0H2. Here through this reaction, 4 protons will be pumped. 4 protons will be pumped from NADH. Okay, so the four protons will be pumped out from the matrix to outer mitochondrial membrane. So not only electron transport is occurring, proton pumping is also taking place. So in the first complex, the flow of electrons is taking place simultaneously, pumping or pushing out of protons also occurs. So for every proton's entry, for every electron transport, protons also will be pumped out from the matrix to the outer mitochondrial membrane. Then the second complex is called as succinate dehydrogenase complex. So second complex is called succinate dehydrogenase complex. Here the source of electrons will be succinate. From succinate it will be donated to FAD. The electrons will be passed to FAD. Then from FAD it is donated to, the electrons will be passed to FES center. Final acceptor will be ubiquinone. So again for second complex also the final acceptor is ubiquinone. From succinate the flow of electron occurs through FAD and FES center. Finally it will be given to ubiquinone. So FAD, finally FAD will be FADH2 will be converted into FAD. So this is in the actually in this second complex, the pumping of proton will not occur because the two protons will be utilized to oxidize this ubiquinone into reduced form of ubiquinone, and other one more 2H plus protons, other 2H plus proton will be utilized to convert this FADH2 into FAD. So here no pumping of proton occurs in the second complex. The third complex is called as coenzyme Q cytochrome C oxido reductase complex. So here the electrons will be, the flow of electron will be from ubiquinone. So ubiquinone will be acting as the source of electron. The final acceptor for complex 3 is cytochrome C, hence the name is coenzyme Q cytochrome C reductase complex. From coenzyme Q the electrons will be passed to cytochrome B, then to FES center. From FES it will be donated to cytochrome C1. From cytochrome C1 it will be passed to cytochrome C. So this is how the flow of electron occurs for this complex 3. So in this the ubiquinone will be converted into the oxidized state, and cytochrome C will be in the reduced state. Through in this third complex also pumping of 4 protons occurs. So totally from first complex 4 protons have passed out, and from third complex again 4 protons, so 8 protons will be passed out through NADH, and for every FADH2 only 4 protons will be passed out. Okay, because in the second complex there is no pumping out of protons. So this third complex also will act as a proton pump. So whenever proton pumping 4 H plus ions are pumped out, it will be associated with generating one molecule of ATP. So through first complex, one molecule of ATP will be generated, and again through third complex, one molecule of ATP, that is oxidative phosphorylation site 2. This will be acting as a site for oxidative phosphorylation. That means it will also generate ATP. Then fourth complex, this is called cytochrome C oxidoreductase or cytochrome C oxidase complex. Here the flow of electron will be from, so the cytochrome C will be the source of electron. From cytochrome C, the electrons will be carried via cytochrome B, cytochrome C1 and FES center. It also acts as a proton pump. So here also from fourth complex also proton pumping occurs. So it will also leads to the generation of 1 ATP. So it will also act as the phosphorylation site. Okay, for third phosphorylation site will be complex 4. So ubiquinone will act as a connecting link between the complexes 1, complex 2 and complex 3, and cytochrome C will be act as the link between complex 3 and complex 4. Both the cytochrome C and ubiquinone are readily extracted from the inner mitochondrial membrane with the help of solutions butanol, salt solution and butanol. We can immediately extract this ubiquinone and cytochrome C. So in this fourth complex, cytochrome oxidase, finally the electrons will be accepted by oxygen. So half a molecule of oxygen will be converted into one molecule of water. So the final acceptor for complex 4 is oxygen conversion into water. So here only out of this 4 protons, only 2 protons will be pumped out from the mitochondrial matrix to outer mitochondrial membrane, and the remaining 2 protons will be utilized to convert this half a molecule of oxygen into one molecule of water. So totally out of this 4 complexes, 10 protons will be pushed out from, if you calculate from NADH, 10 protons will be pushed out, and if you take the number of protons pumped out from FADH2, that is from complex 2, 6 protons will be pushed out. That is 4 protons from complex 3 plus 2 protons from complex 4. So 4 plus 2 it is 6. Whereas if you calculate from NADH, that is first complex donates first complex pushes out 4 protons, third complex 4 protons. So 4 plus 4 8. Then last complex pumps out 2 protons. So totally 10 protons will be pumped for every NADH, and for FADH2 it is 6 protons. So the 6 protons will help to produce 1.5 molecules of ATP, and out of this 10 protons 2.5 molecules of ATP will be generated through oxidative phosphorylation. Okay, this is how the flow of electrons will occur. So complex 1 will be acting as a site for ATP, producing ATP. Complex 3 will be acting as a source for generating ATP, and complex 4 also will produce ATP. So these 3 complexes will be involved in oxidative phosphorylation, whereas second complex will not produce any, generate only ATP. Only electron flow alone will occur through this complex 2. So this is how in the mitochondrial membrane all the 4 complexes will be embedded. Complex 1, complex 2, complex 3 and complex 4. During the electron transport, energy also will be released. This energy will be used to transport the H plus ion across the inner mitochondrial membrane to create a electrochemical gradient. This electrochemical gradient will be utilized by the fifth complex to produce ATP. So whenever protons are pumped out or pushed out from the matrix to outer mitochondrial membrane, so this where the H plus ion load is poor, it will become positively charged. The membrane outside this layer, no, it will become positively charged, and the layer which is losing the proton will become negatively charged. This difference in charge separation is called as electrochemical gradient. So this gradient is very important to generate ATP during oxidative phosphorylation. So during the transport of electron only, this electrochemical gradient is created or generated with the help of the energy released during the transport of H plus ion. So this is how the electron transport chain, the carrier proteins will be arranged in the order of increasing order of redox potential. So this is the value for each substrate metabolite. So according to the from negative to positive, that is this is the standard reduction potential. So this is how the electron transport in the electron transport chain, each molecule will be arranged in the order of their increasing redox potential. So NAD plus, it's having minus 0.32 will be arranged in the first, will take the first, it will be acting as a source of protein, and it will donate the electrons to the next carrier protein, which will have in, okay, so FAD, so it is having minus 0.22, then finally it will be given to cytochrome B, which is having plus 0.07, and ubiquinone, which has got plus positive 0.10. Cytochrome C, the redox potential for cytochrome C will be plus 0.22, and final acceptor oxygen has got the highest standard redox potential plus 0.82. So this is the order of arrangement of metabolite, and the flow of electron also occurs through this standard increasing standard reduction potentials. So this is the standard reduction potential of NADH minus 0.32. It will donate the electron to ubiquinone plus 0.03. From ubiquinone, the electrons will be transported to cytochrome C, which is a plus 0.04. Finally, it will be given to oxygen, which has got plus 0.82. So this is how the flow of electrons occurs from the carrier with low to high reduction potential. So it will be energetically downhill process. Then inhibitors of electron transport chain. So the complex 1, the name of the complex 1 is NADH oxido reductase complex. It will be inhibited by drugs or metabolites like Piercidin, Amitol and Rotenone, rat poisoning. So this Piercidin, Amitol and Rotenone, in the presence of Piercidin, Amitol and Rotenone, the complex 1 will not be able to transfer the electron properly. So electron flow will be inhibited in the presence of these inhibitors, Piercidin, Amitol and Rotenone for complex 1. And for complex 2, that is succinate dehydrogenase complex, malonate and carboxin will be acting as an inhibitors, and antimycin A will be acting as an inhibitor for coenzyme Q cytochrome C reductase complex. And for complex 4, cytochrome C oxidase, the inhibitors will be H2S, carbon monoxide, cyanide and sodium acid. So all these will act as inhibitors which may block the electron flow. So these are the specific sites how it will block the flow of electrons. So in the presence of Amobarbital, Rotenone and Piercidin A, the flow of electron from FMN to FES center will be blocked in the first complex. In the presence of malonate and carboxane, the flow of electron from FES center to ubiquinone will be blocked. So this is a specific site where the electron flow will be blocked in the complex 2. Then the third complex will be blocked with the flow of electron will be blocked from cytochrome C1 to cytochrome C with the help of antimycin A. Okay. Then in the for fourth complex, the flow of electron will be blocked from cytochrome A3 to of a molecule of oxygen. So this will be blocked with the help of cyanide, carbon monoxide, hydrogen H2S and acid. Okay. Hydrogen sulfide and acid. So all these are inhibitors which will specifically block the flow of electron from one carrier to another carrier. Hope you would have understood the electron transport chain process. Thank you for watching this video. Subscribe and support the channel Biochemistry DSP.